Hello everyone, welcome to another episode of Bio-Inorganic Chemistry lecture series. Today we are going to learn about metal management in living systems, particularly in focus with iron storage and transport proteins. First, we will see the significance of iron metal. We know that iron is a very important element present in human body and other living systems. It is required for oxygen transport, it is required for DNA synthesis, and also it is required for electron transport. It is present in hemoglobin, myoglobin, cytochromes and iron sulfur proteins. It is very important to maintain a constant balance between the uptake, transport, storage and utilization of iron, and it is known as iron homeostasis. Moving on, we will first learn about iron transport proteins. The main iron transport protein is transferins. Transferins are non-heme iron binding proteins. They are non-heme, that means it does not contain the iron porphyrin ring, and they are binding blood plasma glycoproteins that control the level of free iron in biological fluids. They are actually blood plasma glycoproteins which are non-heme proteins, and it controls the level of free iron in biological fluids. What is the function of transferrin? It transports iron from absorption centers to the centers of utilization, storage and hemoglobin degradation. They transfer iron from the absorption centers of iron to sites of utilization, storage and hemoglobin degradation. It has key role in erythropoiesis and active cell division. It is produced by the liver cells, in which it is then secreted into the plasma. It is produced by the liver cells and then secreted into the plasma. You can see the structure of human serum transferrin. We have different types of transferrin. This is the structure of human serum transferrin. We will see the structure in detail in the coming slides. Now we are going to see the classification of the iron transport protein, that is transferrins. We have 4 categories of transferrins. The first one is serum transferrin. We have lactotransferrins, ovotransferrins or quinalbumin, and finally we have melanotransferrins. First we will see what are serum transferrins. These are also known as serotransferrin, siderophilin and also 13A metal binding globulin. It plays a vital role in the transport of iron within the circulatory system of vertebrates including humans. It plays a vital role in the transport of iron within the vertebrate circulatory system. Next we will see lactotransferrins. These are generated by the mucosal epithelial cells in mammals. Their function is body defense against infections and has antioxidant, anti-inflammatory and antimicrobial activities. Next we will see what are ovotransferrins or quinalbumin. They are known as ovotransferrin or quinalbumin, which is commonly found in the oviduct secretions. Similar to that of serum transferrin, they are derived from the same gene of serum transferrins. Another category of transferrins is the melanotransferrin. These are also known as cell surface proteins. The first cell surface markers associated with melanomas. The major role is in cell proliferation, migration, angiogenesis and tumorigenesis. So, the most important one is the serum transferrin that we are going to see in detail. Next, we are going to see in detail the structure of transferrin, particularly the human serum transferrin. It has a molecular weight of about 79.6 kilodalton. It contains polypeptide chain which has 679 amino acids. The structure is actually bilobal structure of equal size. In this figure, you can see the two lobes, that is the N lobe and the C lobe. Both are having almost equal size and they are linked by a polypeptide. Each lobe is further divided into subdomains and interconnected by two anti-parallel beta strands. So, you can see that the human serum transferrin contains two lobes, that is, it is having a bilobal structure. You can see it is being connected to the amino acid residues histidine particularly and aspartame, tyrosine, etc. Next, we are going to see the mechanism or the mode in which the ion is transported by the transferrin. The transferrin without ion is known as apotransferrin. The ion binding actually occurs very tightly but reversibly. The affinity of transferrin for Fe3 plus is very high but decreases with decrease in pH. That is, the ion is having more affinity, more strongly affinity to the ion in the 3 plus oxidation state. That is, the transferrin is binding ion 3 plus very strongly and it decreases with decrease in pH. The lobes bind one ion atom with of the 6 ligands. Each lobe will bind one ion atom with the 6 ligands. It contains two specific high affinity ion 3 binding sites. I have already mentioned that it contains 4 ligands, that is aspartic acid, tyrosine and histidine from protein and 2 ligands synergetically from the carbonate anion. Here you can see that the ligands in transferrin, it will bind Fe in the 3 plus oxidation state and it has the 4 ligands histidine, aspartame and tyrosine, and the other coordination is compensated by the bicarbonate anion. Now, we are going to see what are the ion storage proteins. The major sites of ion storage, we know that it is liver, bone marrow and spleen. Actually, ion storage occurs in two forms. It is stored by ferritin and also it is stored by hemosiderin. Initially, iron is stored as a protein ion complex, ferritin. However, with time, the ferritin molecules aggregate to form clusters which are subsequently engulfed by the lysosomes and consequently degraded. As a result of this degradation, what happens is that the hemosiderin that is formed, that is an amorphous aggregate of the denatured protein. We understand that iron is stored in two forms, that is ferritin and hemosiderin, and it is stored as an iron protein complex, and with time what will happen, these molecules aggregate to form clusters and which will finally be engulfed by the lysosomes and it will be consequently degraded and then it will form an amorphous aggregate of denatured protein, that is the hemosiderin. Now, we will see what is ferritin. Ferritin is the principal storage form of iron in the cells. It is a water soluble intracellular protein. Ferritin molecule forms a hollow sphere. You can see it is a hollow sphere which contains about 2000 iron atoms and can hold up to 4500 iron atoms. You can see the capacity of ferritin. It is a hollow sphere which contains, this is the iron metal core, which can store up to about 4500 iron atoms. Now we will see the structure of ferritin. It is having a protein coat and inorganic core, which is suitably encapsulated within the core. You can see this is the core iron, and surrounding it you can see the protein coat. This is the protein coat, and inside you have the mineral iron, or this is the point where iron is stored. The protein molecules are spherical in shape, containing iron atoms in the form of a mineral. You can see the 3D view of ferritin here. It is having a spherical structure and it is having the iron core inside the protein sphere. Now let us see in detail what is the protein coat. Actually, the protein coat is a spherical assembly made up of about 24 polypeptide subunits. Each subunit will contain almost 175 amino acids. In the interior of the symmetrical protein coat, there exists a hollow sphere of approximately 100 angstrom diameter. At intersection of the two subunits, two separate channels are found filled with varying amounts of solvent and iron. These channels have a fourfold and threefold access to allow the movement of iron in and out of the protein. That is, these channels have a fourfold and threefold access to allow the movement of iron in and out of the protein. This is the structure of the protein coat in ferritin. Now let us see the structure of the iron core. That is, this is the iron core. You can see that it is stored in the form of ferric oxyhydroxyphosphate complex. Stored as ferric oxyhydroxyphosphate complex. The formula is, this is ferric oxyhydroxyphosphate complex. And the iron cores are actually unevenly arranged. Only a fraction of iron 3 that binds directly to the protein. I have already mentioned that ferritin will capture iron or have more affinity to iron in the +3 oxidation state. Now let us study the magnetic properties of ferritin. Every iron atom within ferritin molecules contains at least 3 unpaired electrons. That means iron is in the +3 oxidation state and in the high spin form. The internal iron core in the nanodomain contain Fe3+ ions whose individual high spins couple in a way to create a superparamagnet at ambient temperature, and hence this core material is actually superparamagnetic. The core acts as a solid piece of metal having a single magnetic domains. That is, it is superparamagnetic and it will have single magnetic domains. Now let us see the function of ferritin. We have already seen that it is the storage of iron. It provides a reserve of the iron which can be drawn when needed. It protects the cell from toxic effects of free iron. So that is an important use of ferritin because the protein coat will reduce the toxicity of iron and it keeps the iron as a reserve and it will be made available to the body when required. How does ferritin store and release iron? It is controlled by a mechanism and it is a controlled mode of release. That is, I already mentioned that iron is stored in the form of iron 3. So, during the release, what happens? Iron 3 is being converted into iron 2 oxidation state. The iron 2 form will leave through the threefold and fourfold channels in the spherical structure. So, there is a switching during the release from the iron 3 oxidation state to the iron 2 oxidation state. This is how iron 3 plus will move out when required in the form of iron 2 plus. Now, we move on to the next iron storing protein, that is the hemosiderin. So, commonly it is found in the macrophages. So, what are macrophages? We know that it is a type of blood cells that is needed for the immunity, and it is formed during the hemoglobin breakdown. So, it appears to be a complex of ferritin, or we can say it is a denatured ferritin molecule. So, iron in hemosiderin is actually poorly available to deliver iron when required. Iron in ferritin is actually good for the release when required, but hemosiderin is also a storage of iron, but it occurs mainly, or it is formed mainly during the breakdown of hemoglobin molecules. It is abundant following hemorrhage. Okay. You can see the dark hemosiderin granules in the liver cells. Okay, the Prussian blue stain corresponds to the hemosiderin. Now, let us see the formation of hemosiderin. The iron protein ratio in hemosiderin is actually higher than that of ferritin. The insoluble species is formed by the lysosomal decomposition of ferritin. Protease in the lysosome will degrade the protein shell of ferritin. The release iron core will then dissociate and reforms to the amorphous form. So, after the what happens is that there will be degradation of the protein shell, and then iron will be released, and or the iron core will dissociate and it will reform to the amorphous form. This is how the hemosiderin is formed. Now, we will look into the magnetic properties of hemosiderin. It is also super paramagnetic like ferritin. The magnetic susceptibility effects are actually more powerful owing to the large particle size. We say that the magnetic susceptibility effects are more pronounced in case of hemosiderin. Now, we will see the next category of iron storage, that is actually we call siderophores as iron carriers. These are mainly found in the lower organisms like bacteria and fungi. Siderophores are actually iron carriers which are small and have high affinity iron chelating compounds that are secreted by the microorganisms. So, these help the microorganisms to accumulate iron and is also essential for these lower organisms. So, ferric ion is actually poorly soluble and it is necessary for the microbial growth. So, bacteria needed iron and they produce and release siderophores. They function as a natural high affinity iron chelators that enable iron into the cells. They are actually forming an iron chelate complex, and that is how it works inside the microbes. The molecular weight is very low, that is about 4 to 20 kilodaltons. It is produced in presence of iron shortage to scavenge iron and form insoluble iron 3 complexes. It is involved in pathogenic bacteria taking iron from the host proteins because they allow organisms to access iron in the insoluble form. Actually, the microorganisms are unable to biosynthesize high molecular weight complex proteins. So, what they do? They gather iron from the surroundings via siderophores, solubilize and transport iron 3 plus by forming extremely stable octahedral complexes with iron 3 plus, and finally transport it across the membrane. You can see that they are unable to synthesize high molecular weight protein complexes. So, what they do? They gather iron from the through the siderophores and make it, I already mentioned the solubility of iron 3 plus is low, and thus by chelating they solubilize the iron 3 plus and they form stable octahedral complexes. We can see the structure of a ferrichrome complex, it's a siderophore, and transport it across the cell membranes. Based on the ligands used to chelate ferric ion, siderophores are divided into various categories. Siderophores are categorized into catecholates and hydroxamates and carboxylates. This is based upon the ligands that are used to chelate the iron in the 3 plus oxidation state. For example, ferrichrome, this is a type of hydroxamate. You can see this, an example, that is ferrichrome complex. It is a siderophore which is a hydroxamate complex. So, there are different types of ligands that will complex with iron 3 plus, and they are utilized by the lower organisms because they are not capable of producing high molecular weight complexes. Moving on to the summary, we have learned about iron transport proteins and the iron storage proteins. The iron transport proteins are the transference. These are basically non-heme iron binding blood plasma glycoproteins that are capable to control the level of free iron in biological fluids. They transport iron from the absorption centers to the sites of utilization. We have also learned about the iron storage proteins, mainly ferritin and hemosiderin. They are mainly produced at the main sites of storage, not produced, main sites of storage are liver, bone marrow and spleen. We have also learned about the bacterial siderophores, which are the high affinity iron chelating compounds secreted by the microbes such as bacteria and fungi. They help these organisms to accumulate iron. Thank you.